Hello, welcome to Mr. Faye's Science Basement, the back porch edition, where today we're going to be talking about molecular evidence for common ancestry. If you take a look here, you're going to see the notes that I supplied in the Google Classroom. You can make a copy of them and write on them digitally. You can print them out and take notes with pen or pencil, cut them out and put them in your scrapbook, or you can take your notes freestyle. That is up to you. In front of you right now, you have a DNA test. Anytime DNA is left at the crime scene, or you hear of something called a paternity test, or even with things like Ancestry.com, 23andMe, a lot of them work in a very, very similar principle, where DNA is cut up and then the pieces of DNA that are separated are compared to each other to help identify it. So this right here would be a great example of something called an electrophoresis gel. In other words, different samples of DNA are dropped into these little reservoirs that you see up here. Then they're processed in a way we'll talk about shortly, and they're used to compare similarities. And if those similarities are abundant, then that means that you have DNA that's closely related. All right, so here's the short version as to how it works. An electrophoresis gel is exactly that. It's a gel. It kind of looks like Jell-O, actually. This little blue part you see is poured into a mold and has these little wells in it. Each one of these wells is going to be treated with a different sample of DNA. So you might have crime scene DNA and maybe 3 suspects' DNA put into these wells. Now, before the DNA is put into the wells, they're actually cut up into pieces. So the crime scene DNA will be treated with something called a restriction enzyme. And that restriction enzyme cuts DNA sequences in specific places. You then take each one of the other individual's DNA, treat it with the same restriction enzymes to cut them in specific places, and then you allow them to progress through the gel. As they progress through the gel, they're going to leave bands or patterns. If that DNA is closely related, those bands will be the same. If they are not closely related, then those bands will be different. Now, just a real quick aside, the way that they actually move is relatively simple. The gel itself is charged with 2 different electrodes, a negative up here and a positive up here. The DNA is treated in such a way that it is negative, and it's actually drawn through the gel to the positive. Big pieces of DNA move very slowly through the gel, so the big pieces will be deposited back here, but the smaller and smaller and smaller pieces will be deposited further along the electrophoresis. So again, if this is the crime scene DNA, whichever has the bands that match up the most closely could be the perpetrator. Okay, so this is pretty interesting. Human DNA can be compared to different human DNA, and you can definitely tell those differences. So you specifically have your own banding pattern. However, all humans have over 99.9% of their DNA identical, and those changes in the banding is actually going to be pretty minimal if you're looking at the same species. So all humans really do share about 99.9 plus percent. And when you're looking at crime scene DNA, you're actually focusing in on less than a few tenths of a percent of that DNA difference. Now, something that's kind of interesting is if we put you and let's say a sibling into that same electrophoresis gel band, we'd be able to tell whether somebody were brothers or if they were cousins or if they were second cousins or not related, because you actually have more DNA in common with your direct sibling than you would with a cousin. And if you have 2 people that are unrelated at all, there'll be even a bigger difference. But all of those differences still lie in less than 0.1% of that DNA sequence, which is crazy when you think about it. DNA banding, like in the electrophoresis gel that you saw, can actually be used to tell how closely related different species are to each other. So for example, if you had an unknown species of plant, you cut up its DNA and sent it through an electrophoresis gel, you might find that as a banding pattern. Then you could take 4 other species of plants, A, B, C, and D, do the same thing to them, and whichever banding pattern lines up most quickly will show you how closely related they are. And if we take a look, it looks like C is pretty closely related and possibly D, but I think C might be the most closely related to A. I'm sorry, to the unknown. So even though these are different species, realize we can look for similarities across species to see how closely related those species are. Okay, so here is where it gets super freaky. If you're looking at humans, All humans, we share about 99.9% of our DNA in common. But if we look at human DNA compared to other species, we do have DNA in common, or at least more in common with species that we're more closely related to. So, for example, if I pull up a chimpanzee, we actually share about 96% of our DNA with a chimpanzee. Now, when you think about it, characteristics are similar, so we could definitely see some relationship there. But that's a lot of DNA to have in common if you really think about it. Quick misunderstanding moment right here. This does not mean that people came from monkeys or people come from chimpanzees. It suggests that people and chimpanzees in the past could have a common ancestor and are related to each other in some way. What even is weirder is if you look at some other animals. For example, if you take a look at a human and a cow, we share about 80% of our DNA. If you take a look at a human and a fruit fly, we share about 61% of our DNA. So, as you can see, most life is pretty closely related on the planet. But if we were to look at all these animals that we gave examples of, we're most related or most closely related to the chimpanzee. So these molecular similarities are not just limited to DNA sequences. You can also look at the amino acid sequences in various proteins. And a great example of this is taking a look at a portion of hemoglobin. And as most of you know, hemoglobin is an enzyme inside your red blood cells that carry oxygen. If we were to take a look at a portion of a hemoglobin molecule, humans have 146 amino acids in common with each other. In other words, we have 146 amino acids in this portion of the hemoglobin chain, and they're all the same for people. However, if you go over and take a look at the rhesus monkey, a pretty close relative, we'll find that there are only 8 of those amino acids that are different. So the basic amino acid order in the hemoglobin is pretty much the same, with 138 being the same and 8 being different. As you move further and further away in relationship, you're going to notice that we have 32 different with a dog, 45 different in our hemoglobin chains with a bird, and 125 different when you take a look at a fish. And that makes a lot of sense too, because fish lives in a completely different niche, having to extract oxygen from the water, which probably has to change the structure of the hemoglobin quite a bit. So again, not just specific to DNA. Sometimes we're talking about amino acids and proteins too. So, looking at molecular evidence that in DNA and the amino acid sequence of proteins, we can start to build a pretty complete picture of relationship. So if we take a look here, we're going to see that like rodents and primates, they would have a higher percentage of their DNA and amino acid sequences in common. Therefore, you don't have to go back as far to find some sort of common ancestor point. And it kind of makes sense too, because if you think, well, both of these have the DNA code to make things like hair and fur, and that's going to give you some similarities there. As you back up this chain here, you're going to find that you have less DNA in common between rodents and primates than you do with reptiles and birds, but reptiles and birds have a pretty high similarity in protein and DNA content. But they can be traced back to this point here, which means that there is some common ancestry. In other words, these things all have some DNA in common and have even less DNA in common with things like amphibians, and yet again, even less with things like fish. So, looking at DNA sequences, the percent that you share, looking at amino acid sequences and proteins, you can start to paint a pretty rich picture about this idea of common ancestry. How far do you have to go back to find a common ancestor between species? Hopefully that helps out. If you have any questions, feel free to ask in an upcoming Zoom conference or as a comment in the classroom. See ya.